The tutorial introduces two experiments presented in my toolkit, my collaboration with bacteria for paper production. The toolkit includes agar plates, scales, a microscope, and ingredients for the experiments. The experiments explore bacterial cellulose, which differs from traditional plant cellulose used for paper, paperboard, and cellophane production. The experiments will give an idea about symbiotic relationships between microorganisms and in a broader context between chemical and organic elements which while interacting evolve into new configurations. In this experiment, I will show how to grow SCOBY, a symbiotic organism of yeast and bacteria, which, if dried out, could be used directly as paper. The experiment could be also interesting from the perspective of consumption, because the grown SCOBY produces fermented tea and could be used as a beverage. Or if fermentation process takes a longer time, it could be used as vinegar for different meals. In the experiment, we will use a electric stove, a strainer, a sheet of baking paper, cloth, a jar, scale, and we will use different ingredients. SCOBY, a sample of SCOBY and kombucha tea, tap water, sugar, and a green tea.
Boil the water in order to kill unwanted microorganisms. Add a tea bag. Leave it in for at least 10 minutes to steep and throw it away afterwards. Add sugar and mix the solution thoroughly until the sugar dissolves. Allow the tea solution to cool to room temperature and add it to the sample of the SCOBY with kombucha tea. Place the jar in a safe place. It will take 2 to 3 days to see the start of the formation of the new SCOBY. After around 10 days, your SCOBY floating on top of the tea should reach 3 to 5 millimeters in thickness. Use a strainer to strain it from the unwanted bacterial pellicle. The brewed kombucha tea should be ready to drink. Put the grown SCOBY on the baking paper and let it dry for a couple of days until the pellicle is ready to be used as bacterial paper. For further experimentation, use black tea, red beet juice, or other natural ingredients. You may also want to experiment with growing the pellicle for different lengths of time or in differently shaped containers.
The experiment introduces the isolation of Acetobacter bacteria from the SCOBY culture. If grown, Acetobacter could be further used for cellulose production. The experiment also shows how to inoculate bacterial species from a colony of bacteria. It could be interesting for further research and analysis of living organisms. To prepare about 15 milliliters of acetobacter medium, we will use following equipment: an electric stove, a knife, a marker, a paperclip, a jar, agar plates, a pipette, and scales. We will also use following ingredients: a sample of SCOBY with kombucha tea, glucose, peptone, yeast extract, disodium phosphate, agar, citric acid, and distilled water. We will also need some alcohol.
Mix the ingredients as outlined. Boil the mixture in order to sterilize it. Sterilize the agar plates provided. Pour the medium into each of them and leave it all to stiffen. Cut a couple of square millimeters of SCOBY out of the culture provided. Put the pieces of SCOBY into the empty jar. You may add some kombucha tea as well. Add some distilled water to the jar. Shake the mixture for a couple of minutes so the microorganisms dissolve into the water. Pour a drop of the solution onto the first plate. Spread the solution with the paperclip. Prepare a 1 to 10 solution of dissolved microorganisms. Use a pipette and the empty jar. Pour 1 drop of the solution onto the second agar plate. Spread it with the sterile paperclip. Repeat the 1 to 10 dilution with the diluted solution and pour the third agar plate. Repeat this process with the fourth and the fifth agar plate. This will ensure that you identify the Acetobacter colony grown from one single bacterium. Leave the sixth plate unchanged. Label your agar plates with a permanent marker for your records. Flip the agar plates upside down and leave them at room temperature for the next 2 to 3 days. After those 2 to 3 days, inspect your agar plates. In our case, the first agar plate shows diverse colonies of bacteria that spread all around, and one colony of yeast that developed on the right side of the plate. The second plate has 3 colonies of Acetobacter in the middle of the plate. The third plate has only 1 colony on the right side. The fourth and fifth plates have no bacteria and no yeast, which means that the sample with kombucha had been diluted too much. So the most successfully isolated bacteria are on the second and third agar plates. We will use the colony on the third plate for the new inoculation. Use the sterile paperclip to take the visible sample of the Acetobacter bacteria from one colony. Inoculate it into the sixth agar plate by carefully placing it onto the gel. If the experiment was successful, it should be clean. For further experimentation, try isolating the Lactobacillus bacteria or Candida fungi found, for example, in saliva. Use medium appropriate for the microorganisms you want to isolate.